Hello friends, I am Dr. Ujjwala Lande, Assistant Professor, Department of Zoology, Sri Shivaji College of Arts, Commerce and Science, Akola. Today we are going to discuss about the topic cytoskeleton. So friends, let us understand what is cytoskeleton. Cytoskeleton is a system of filaments or fibers that is present in the cytoplasm of eukaryote cells. So here you can see how thin filaments are there present here, and it contains, this is a complex network of filaments of and as well as the microtubules, and this forms the cytoskeleton. This provides a structural support to the cell as well as it helps in cell motility and regulation. So you may say that all the constituents of the cytoskeleton, it helps in maintaining the cell's shape. It is also responsible for locomotion of the cell as well as movement of various organelles within the cell. You can see here, this is the internal structure of a cell, and in this internal structure you can see this part where my cursor is there. There you can see that there are fine filaments or fine fibers that you can see, and these fine fibers, they constitute the cytoskeleton. These fibers are microtubules, intermediate filament, microfilament. So these are extremely minute complex interactive network which are formed by these three types of filaments, and they are concerned with the structural framework, orientation and distribution of cell organelles, movement or within the cell, and maintenance of the shape of the cell. So it is also the site of anchoring mRNA and facilitating their translation into proteins. Therefore, it is also referred as cytomusculature. And we must note that cytoskeleton is absent in bacteria. In this slide, you can also see that there are two cells you can see, and in this the yellow color is showing the cytoskeleton. So again we are repeating here that the cytoskeleton, it is to create the shape, to change the shape as well as to allow the movement. So that is, it is providing mechanical support to the cell, it is maintaining the shape of the cell. These fibers, they act like geodesic dome to stabilize and balance opposing forces. It also provides anchorage for organelles. It is very dynamic. What exactly it is, we are going to understand in the following slides. Dismantles in one spot and reassembles in another spot to change the shape of the cell. So, main kinds of fibers of cytoskeleton are three, that is the microfilament, the intermediate filament and the microtubules. So, about these three types of filaments that we are going to study in the following slides. So, here you can see the picture. Here you can see the intermediate filaments that you can see here. These are the intermediate filaments, which the brown structure that you can see. Then this is the microtubule, the green color. This is the enlarged structure, that is of the micro. It is the largest one you can see compared to three. And these fine filaments are called as the actin filaments. You can see how thin they are. How thick are the microtubules, and these are the intermediate filaments that you can see here. So this is the cell membrane, and how the intermediate filament is making the framework and giving shape to the cell. In this slide also, you can see the same structure, that is in a different form. You can see the plasma membrane is shown from both the sides, and in between you can see the green color structure, how it is building up a framework to give shape to the cell. This is the intermediate filament. All these are the intermediate filaments where you can see my cursor, and then here near the plasma membrane you can see the thin fibers. They are the actin filaments. They are made up of actin monomer. These are the fibrous filament made up of fibrous subunit. And then you can see here the tubules, the blue color tubule, this one and this one. These are the tubules, and these tubules are made up of monomers, two types of monomer, that is the beta tubulin as well as the alpha tubulin. These two colors are shown here. Two colors and two different types of tubulin are shown. One is the beta tubulin and second one is the alpha tubulin, that is shown over here. Now we are going to understand one by one all the three types of filaments. So in the previous slide we have seen that the actin filament or the microfilament, they are the thinnest filament that is present in the cytoplasm of eukaryotic cell, and they are giving shape and enabling the movement of the cell. So actin filament occurs in a cell in the form of meshwork of bundles of parallel fibers. They help determine the shape of the cell and also help it adhere to the substrate. The constantly changing arrays of actin filaments help move the cell and mediate specific activities within it, such as cell cleavage during mitosis. Here you can see that these are the globular filaments. These are the globular proteins you can see. So these small structures that you can see, that is the globular protein, and these globular protein, they polymerize to form long filament. These are the small monomers or the subunit. Because each actin subunit faces the same direction, therefore the actin filament is polar with different ends, termed as the barbed end as well as the pointed end. And these are the globular actins. They are called as the G actin, and once the filament is formed, we called it as filamentous actin, that is F actin. So this protein is abundant in all eukaryotic cell and has been extensively studied in muscle cells. So the polymerization of actin, here you can see the polymerization of actin is shown here, and this process by which the monomers of the proteins, they combine to form actin filaments, and this process is called as nucleation. This process is called as nucleation. So nucleation occurs when a group of 3 or more actin monomers spontaneously or otherwise group together, forming a base onto which actin monomers can attach. The polymerization of actin does not form a single linear strand. It forms rather an active filament consisting of the double helix of linked monomers that you can see here. Such an arrangement is much more durable than a single linear strand would be. So that is shown here in these two pictures. So actin polymerization is a reversible process, meaning the actin filaments can be broken down into individual units of actin. This makes for a very dynamic process, as actin filaments can polymerize and depolymerize quickly at different locations throughout the cells. And various chemical changes in different parts of a cell can promote polymerization or depolymerization. So such actin filaments can be assembled and disassembled quite rapidly based on particular needs of the cell. Now the functions of the microfilament. The basic function of the microfilament is to form dynamic cytoskeleton, which gives structural support to the cell and links to the interior of the cell with its surrounding. Forces acting on the actin cytoskeleton are translated and transmitted by signaling pathways to convey information about the external environment. These filaments also allow cell motility, that is, formation of filopodia or lamellopodia, with the help of which these structures can move. Now, during cell division or mitosis, intracellular organelles are transported by motor proteins to daughter cells along actin cables only. In muscle cells, actin filaments are aligned and myosin proteins generate forces on the filaments to support muscle structure. These complexes are known as thin filaments. In non-muscle cells, actin filaments form a track system for cargo transport that is powered by non-conventional myosins such as myosin 5 and 6. Non-conventional myosin use the energy from ATP hydrolysis to transport cargo, such as vesicles and organelles, at rates much faster than the diffusion. Here in this slide, you can see the drugs that affect actin filament. So this point or this table is very important because in CSIR net exam, questions are many a time is asked on the drugs. If a drug is given and then you have to identify its effect or its role. That type of question is asked. So mainly these 5 types of drugs that are studied, that is cytochalcin D. So what cytochalcin D is doing, it binds plus end of the actin filaments and prevents elongation. Also, it is playing a role in depolymerization of microfilament. Then latrunculin. So it binds G-actin monomers and prevents them from polymerizing into filament. That is, it prevents formation of filament. Phalloidin binds tightly all along the sides of actin filaments and stabilizes them against depolarization. Means this is preventing depolarization. This is preventing polymerization. So this way you have to remember that which drug is having what effect. Jasolacinoid induces actin polymerization by stimulating actin filament nucleation, as well as it also stabilizes the F-actin. Svinholite, it serves the filament. So this is all about the microfilament. Now we will move on towards the next filament, that is the intermediate filament. So as the name suggests, they are so named because they are thicker than the actin filaments and thinner than the microtubules or muscle myosin filaments. Intermediate filaments are a very broad class of fibrous proteins that play important role as both structural and functional elements of the cytoplasm. The size ranges from 8 to 12 nanometers and they are composed of two anti-parallel dimers that we are going to see. So you can see here in this diagram, a monomer is shown. In this diagram, two anti-parallel dimers are shown, and in this diagram you can see that there is a tetramer shown. So structurally, all intermediate filaments start from a fibrous unit, subunit. So you can see this is a fibrous subunit. This two of such units, they coil around one another to form a dimer that is called as protofilaments, and these protofilaments then interact to form a tetramer, which are considered the basic unit of intermediate filament construction. So you may say that this one is the protofilament and this protofilament is the basic unit of intermediate filaments. Now in this picture again you can see the same thing is shown, only thing is that the helical region is shown here. It is a protein, it is a protein, obviously it has got the N terminal and C terminal. This is the C terminal and this is the N terminal. So in the next here you can see the two alpha helical region in the monomer, they are coiled around one another to form a dimer. Still they are having one N terminal and another C terminal. In the next diagram you can see that in one here you can see the staggered tetramer of two coiled dimers. You can see this is the one dimer and this is the second dimer, and these two dimers, they have formed a tetramer, and here you can see that the two tetramers are packed together in the diagram. This is what is shown here. In this way you can see how the tetramers, they are forming the filaments, the long filaments in the cell, how they are formed over here. The same diagram, the same things are repeated. Here you can see the dimer is formed, the N terminal and the C terminal. In this diagram, one dimer, this is the second dimer, and how the dimers are arranged to form the protofilament, and this protofilament is forming the 8 to 12 nanometer wide intermediate filament. That is what shown over here. Now in this picture, again the same things are repeated. Here in the lowermost diagram you can see this is the basic monomer of an intermediate filament. This is the N terminal, that's what is called as the head. The middle part is called as the rod, as well as the C terminal, that is called as the tail. Now what happens in the first step? In the first step, two monomers form parallel dimer. That means you can see both the heads are toward one end. This is the dimer. Now in the second picture you can see, or the second step you can see, two dimers, they arrange themselves in anti-parallel alignment, that is side by side, and form a tetramer. So here this way you can see two heads, this way you can see two heads. So now what happens? The polarity is lost here. In the first diagram there was head as well as a tail, therefore there was polarity. In the second diagram you can see they have lost the polarity because they have aligned themselves in anti-parallel way. Now, the tetramers align to end to form the protofilament that you can see here, and the pairs of protofilaments associate laterally to form a protofibril, and 8 protofibrils wind up to form a rope-like 10 nanometer thick intermediate filament that you can see, and this is the way how the intermediate filament is made. The same things are repeated over here in a different view so that you get the idea how they are formed differently. Now come to the functions of intermediate filaments. So intermediate filaments, they function as tension bearing elements to help maintain cell shape and rigidity and serve to anchor in place several organelles, including the nucleus, desmosomes. Intermediate filaments are also involved in formation of the nuclear lamina, a net-like meshwork array that lines the inner nuclear membrane and governs the shape of the nucleus. Here you can see different types of intermediate filaments. There are 4 types of intermediate filaments: type 1, type 2, type 3 and type 4. So type 1, they are the acidic and basic keratins. They are present in hair and nails. Type 2, they are vimentin, cinnamon and desmin. They are present in muscle and glial cells. Type 3, they are neurofilament proteins present in neurons. And type 4, they are the nuclear lamins A, B and C, and they are present in nuclear lamina of all cells. Here this is the point where you may get a question in CSIR NET exam on the intermediate filaments. Now come to the next one, that is the microtubules. The picture itself is showing us that how thick this filament is. So microtubules are the thickest fibers, are hollow rods of about 25 microns in diameter, and microtubules are made up of globular protein, that is tubulin, and they grow or shrink as more tubulin molecules are added or removed, just like we have seen in case of microfilament, the way the globular G protein was added or it was removed. So these microscopic hollow tubes, they are formed of alpha and beta tubuli, that is, they form the cell's skeleton, a network of protein filament that extends throughout the cells, give shape to the cell, keeps the organelles in place, and these are the largest structures. They have roles in cell movement, cell division, and transporting material within the cells. Now let us see the structure of microtubule. In this structure you can see that microtubule helical structure is shown. As we have said earlier, microtubules are hollow cylinders. They are made up of repeating protein structure, especially dimers of alpha and beta tubuli. So here you can see this is a dimer made up of alpha as well as beta tubulin. This green color is the beta tubulin and this light yellow or pale color is the alpha tubulin. So this is a pair, or this is a dimer. This dimer is going to build up the microtubule filament. Just like in case of microfilament, we had said only the G protein was making up the filament. In this case, this dimer is going to make the protofilament. In this diagram, you can see that this is the plus end, this is the minus end, this is alpha tubulin, this is beta tubulin. Now how they are added? So to the plus end, these dimers are added and polymerization takes place. Whereas the minus end, the dimers are removed and that is called as depolarization. And if you see the cross section, you can see the microtubule appears like this. So that is the 13 protofilaments arranged into a cylindrical pattern to form microtubules, and microtubules are constantly assembling and disassembling. This I have shown here via the addition and removal of the dimers. That is the reason they are said to be in a state of dynamic equilibrium, because their structure is maintained even though the individual molecules themselves are constantly changing. And the structure, you can see that microtubules are polar molecules with a positively end, charged end, that is the plus end, that grows relatively fast, and a negatively charged end that grows relatively slow. So the protofilaments, they arrange themselves parallel to each other in a microtubule. So the positive end of the microtubule always has beta subunit exposed, that you can see here. The beta subunit, here you can see the green color, that is the subunit, beta subunit is exposed, while the negative alpha subunit is exposed towards the negative end. So here you can see at the red color, this is the negative end, so the alpha subunit is exposed here, and towards the positive end, the green color, that is beta subunit tubular, is exposed here, and therefore it has got a polarity, and this polarity allows the microtubule to assemble in a specific way and function very correctly. Now come to the functions of microtubules. The functions of microtubules are giving shape to the cell and cellular membranes, cell movement, which includes contraction in muscle cells and more, transportation of specific organelles within the cell via microtubule roadways or conveyor belts, that you can see here, the microtubule, how they are forming the roadways for vesicle to move. Mitosis and meiosis, movement of chromosome during cell division and creation of mitotic spindle. Here again we come to the important part, that is the drugs that affect microtubules. So vinblastine, what is the effect of vinblastine? It binds to beta tubulin to prevent polymerization. Vincristine binds free tubulin to prevent polymerization. Taxol binds to and stabilizes microtubules, preventing their depolarization. Colchicine, it binds tubulin subunits to prevent polymerization. Colcimide binds tubulin subunits to prevent polymerization. And here you can see a table that I have made. The table is showing all the comparison is shown over here in the table. The properties of microfilament, that means in a nutshell you get all the things from this table, and if you go through the table very thoroughly, all the slides that we have studied up till now, you are able to study in a nutshell. Hope this lecture is useful to you. Thank you so much.